Ladies and gentlemen, could I ask you to take your seats, please? I have just a couple of announcements. I have an announcement for Mr. Douglas George. Please go to the registration desk in the lobby area for an important message. This is Mr. Douglas George. Okay. And also, you probably noticed that it's raining out today. All right. And those of you who've been with us before know that we usually have coffee and goodies in the area outside here. In view of the weather, we will have coffee and punch in the Lund Forum, which is the building just to the north of us here. So at the conclusion of this session, we'll move into Lund Forum for coffee. You can also get coffee in the Jackson Center, in the Marketplace Lounge, and also the bookstore will be open at that time too. Okay. Now it is my pleasure to introduce to you my colleague in the biology department, Dr. Joel Carlin, who will come up and introduce our next speaker, Dr. Svante Pääbo.
Well, welcome back, everyone. A Nobel Prize-winning biochemist once said that research is to see what everybody else has seen and to think what nobody else has thought. As a doctoral student in molecular immunology at Uppsala University, a young Svante Pääbo applied the technology of viral genetics to something that everybody else had seen: Egyptian mummies. Fearing that his PhD advisor would not like any distraction from his dissertation, he worked nights and weekends until he had successfully extracted and decoded DNA sequences from an infant boy who lived 2,400 years ago. This is no mean feat. Chromosomes are long, protein-covered polymers that, after death, suffer from the breakage of chemical bonds and the formation of new chemical bonds with sugars and amino acids. The result can be a fragmented, cross-linked collection of broken chromosomes that are difficult to extract without further breakage. At the time, the traditional method of obtaining a readable genetic sequence would require the use of bacteria to accept and replicate the DNA. Instead, he used the polymerase chain reaction, a method of rapidly and reliably making large numbers of DNA copies from very few original molecules. Suddenly, the chromosomes of the dead could be read and understood, and his publication became the feature article in the journal Science, all before finishing his doctoral degree. Since then, he has investigated the genetic secrets of extinct giant birds, extinct bears, and other animals. His laboratory developed a method to break cross-links between DNA and other molecules, allowing them to amplify and sequence DNA from, of all things, fossilized sloth feces that had been laid down by a now extinct mammal 10,000 years ago in Nevada. Not only did they recover extinct sloth genes, but also genes from 6 species of plants that the sloth had eaten. What once was a fossil curiosity now tells us about ancient climate and vegetation patterns. Other techniques were developed to recover gene sequences from wild grass seeds domesticated by humans in Central America. Such information allows us now to ask important questions facing modern agriculture. What proportion of our modern corn plants contain the same variations for kernel size or starch manufacture that were found in these original maize? And as we plant more and more corn with less and less genetic variation, what traits of survival may have been lost along the way? The societal impact of his work helps us understand not only ancient organisms, but that which makes us so very human. He discovered that a single point mutation in the FOXP2 gene can cause severe grammatical and linguistic impairment. FOXP2 is present in many vertebrates, but humans have 2 copies that have been the subject of significant natural selection. Thus, changes in select genes, and perhaps more importantly, when these genes are expressed, are tightly associated with our cultural separation from the rest of the animal kingdom. Today, he is discussing his investigations of Neanderthal genomics. The Human Genome Project has greatly increased our understanding of humanity, or at least of modern humans. Where Neanderthals fit into our idea of humanity has been a question of long debate. Were they competitors, mates, victims of, or just disinterested witnesses to the rise of modern Homo sapiens? These questions I will let our speaker address. He is the author of 216 scientific publications which have been cited in the past decade alone by 9,000 research articles. That's more than me if you want to look that up. Since he first worked nights with mummies, he has emerged as both founder and leading force in paleogenetics. This field blurs the lines distinguishing genetics, linguistics, zoology, and archaeology. He has been counted as one of the 100 most influential people in the world by Time magazine and has been honored by the National Scientific Academies of Sweden, Germany, Belgium, the United States, and China. Ladies and gentlemen, it is my very special privilege to welcome to the stage Dr. Svante Pääbo.
Well, dear friends, dear Joel, let me start out then by thanking you all for the invitation to come here to Gustavus and to the Nobel conference. Coming here is not only intellectually stimulating to me due to the talks we get to listen to, it also strangely feels a little bit like coming home, although I've never been here before. Since I'm a native of Sweden but have lived abroad for many years, so when I come here and see all these Swedish flags and all the Swedish paraphernalia in the guest house, it feels a little bit like I finally arrived back home again. So what I wanted to do is to just start out by reminding you all a little bit about what you are all very well aware of, and that's the fact that since about 5 years now, we know the DNA sequence, the 3 billion base pairs or so, that make up the entire genetic material, the genome of humans. And that genetic information are stored on the chromosomes, which exist in the cell nuclei of almost all the cells in our bodies. And as you also are very well aware, the genetic information is stored in the form of the very famous double helix, and it's stored in essence twice, such as, let me try this out then, that when you have, this is too complex, when you on one strand have a C, you always have a G on the other. Whenever you have an A on one strand, you have a T on the other, and so on. And this is important since whenever a cell divides, and especially when that happens in the germline where a new generation is formed, there are enzymes in our cells that unwind the DNA, make the strands come apart, and other enzymes that then synthesize new strands using the old ones as a template. In a process that is almost perfect, always putting in complementary bases, as we say, a G opposite a C and A opposite a T. However, nothing is of course perfect, especially not in biology. So now and again, an error is made. And if enzymes don't correct that rapidly, that may then be incorporated as a difference in the DNA sequence in the next generation. And we can pick that up if we study the DNA sequences of different people, for example, in this room. So if we sequence a piece of DNA from one chromosome in one individual and compare it to another chromosome in another individual, every 1,000 or 1,500 bases or so, we discover a difference between these two individuals. If we take the same segment of DNA from a chimpanzee, we will find more differences, about 1 every 100 bases or so. And so what molecular evolutionists do is then to use this information to reconstruct the history of this piece of DNA as well as we can, using the best models we have for how these mutations accumulate in our DNA. And we generally depict the result of such a reconstruction in the form of a phylogenetic tree. In this case, it's very simple. the two humans trace their history back for this part of their genome to a common ancestor quite recently. And quite a bit further back is there a common ancestor shared also with the chimpanzees. So you can then start using this approach to reconstruct history. You can ask questions such as, who is our closest relative from a genetic perspective? Is it the bonobos, the pygmy chimps, or the common chimps, or the gorillas, or the orangutans in Asia? So you then do that by taking, for example, a piece of our genome, say apart 10,000 base pairs, in one case here, on the X chromosome, and sequence it in one chimp, one bonobo, one gorilla, one human, and so on, and estimate such a phylogenetic tree. And you will then find that on average, for most parts of our genome, humans trace their ancestry back to a common ancestor shared with the two forms of chimps. A bit further back is there a common ancestor shared with the gorilla, and quite a bit further back also with the orangutan. And since these mutations rain down on our genome approximately as a function of time, we can translate these differences we observe in DNA sequences to approximate times to common ancestors for these DNA sequences. And in the case of humans and chimps, it's in the order of 5 to 7 million years ago, to the gorilla a bit more, and orangutan quite further back in time now. But as you notice when we go through this is quite an indirect way to study our evolution. It is using extant species, looking in their DNA, trying to reconstruct what has happened in the past, using models and inferences. What we would like to do in a dream world where we could have everything we would want would of course be to go back in time, to actually look at DNA sequences from our ancestors or from close relatives which are extinct. So what I want to describe to you are our first baby steps to try to do that, to actually do what paleontologists do, to go back and try to retrieve old DNA sequences. And as Curtis sort of pointed out, this goes back now over 20 years, when we started looking into preservation of DNA in ancient Egyptian mummies, which then go back 2,000, 3,000, 4,000 years, and often look very well preserved when you look at them macroscopically. However, if you then look in the tissues in such a well-preserved mummy, it's often quite a depressing undertaking. So this is a contemporary human muscle that had been air-dried to in some way imitate mummification and then rehydrated and processed for microscopy. You nicely see the muscle fibers with the contractile proteins making striations in them. You see the cell nuclei where the DNA would be. If you look in a well-preserved mummy like the one we looked at on the last slide, it looks very different. You barely see there are muscle fibers here. You see no intracellular structure whatsoever. It looks very degraded, and there is no DNA preserved here. However, fortunately, it's not always the case that things are this depressing. So this is the first mummy, which was where we did see evidence of DNA, a sample from the leg here, 2,400 years old, as we heard. And if you look by microscopy in the upper layers of the skin, in the basal level of the epidermis here, you see things that look like cell nuclei. And indeed, you can stain them with dyes that bind to DNA. And you can then go on to extract the DNA from such tissues, just like we would from any contemporary sample. And the first thing you will then notice is that the DNA degraded to short little pieces. This is an analysis where you let your DNA migrate in an electric field. The smaller the pieces are, the further down will they be on this picture. A contemporary DNA would be tens of thousands of bases up here. And you will see that these DNAs extracted from archaeological and paleontological remains are really small, 100, 200 bases. And they're also arranged here from left to right, going from younger to older. So you will see that irrespective of how old they all are degraded approximately to the same extent. This starts out by a 4-year-old piece of a sausage we happen to have in the lab. It goes on to 120-year-old zoological specimen, Egyptian and Peruvian mummies, a few thousand years old, and a 12,000-year-old ground sloth. So this degradation probably happens rapidly after death, actually, as enzymes leak out in our cells as we die and chop up the DNA in small pieces. But there are also other forms of damage in this DNA, chemical damage. If you, by mass spectrometry, look at the DNA, you will find modified forms of bases. Many of them the result of either hydrolytic processes, so spontaneous processes that happen when water is present, or oxidative damage from oxygen radicals from the atmosphere. So your issue then is that if you want to study modern DNA, you have nice, long, nice pieces here. For the ancient, DNA is degraded to short pieces and have these chemical modifications which may cause errors in your sequences. So if you want to look at a longer segment of DNA, you have to retrieve short overlapping pieces and reconstruct the longer part here and still deal with these chemical modifications. So because of this degradation and all these problems, most work in this field has concentrated on one tiny part of our genome that's actually not in the cell nucleus, but outside in the mitochondria and exists in hundreds of thousands of copies per cell. And this little genome has the advantage that there are many copies of it, so you can easily then retrieve it from archaeological remains. And the problems you have to deal with is, as we've said, degradation, damage, and quite important, as we'll discuss in a minute, contamination of your experiments with modern DNA. So all these problems have led to criteria that we and others have tried to promote, you should adhere to, that all have to do with reproducibility of your results, that you yourself should be able to repeat them, and others should be able to repeat them, and chemical analysis to make it reasonable that things have survived in a specimen. What can you then do with this? How far back in time can you go? Well, what is now very routine is to use zoological collections that have been collected over the past few hundred years and exist by the thousands and thousands in museums. Recently extinct animals, such as here the marsupial wolf from Australia that became extinct around 100 years ago or so. The ground sloth that we heard about that disappeared around 10,000 years ago. Mammoths and mastodons that go back 40, 50,000 years, some of the samples. And so one question that often comes, how far back can we imagine we could go in the future? Well, I think, could we imagine to do dinosaurs, for example? Well, I think that will always remain impossible, because if we look on a sort of extrapolation about what we know about the chemical stability of DNA when there are even traces of water present, you can see that if we start with 10 to the 12 DNA molecules, which may be how many mitochondrial molecules there is in a gram of tissue, and see how rapidly they become degraded when water is present, and the last molecule of those 10 to the 12 will disappear before 1 million years ago. So I think that, for example, inclusions in amber that are beautifully preserved and go back many millions of years, unfortunately, it's not possible to retrieve DNA, and even if we found a dinosaur in amber, it would not be possible. But so within the last few 100,000 years then, there's still quite a lot of interesting things you can do. This is just going to 2000, the number of extinct species for which believable DNA sequences have been retrieved. There are many, many more now. But our special interest then in our lab and here today are the Neanderthals that we already heard mentioned by Curtis before lunch. So, as we also heard, Neanderthals, they evolve in Europe and Western Asia. Depending on how you define a Neanderthal, the first traits may go back as much as 400,000 years, and they then become more and more typical. And then modern humans appear, as we heard, between 100, 200,000 years ago, first in Africa, then spread out of the world and exist until the present day. Neanderthals disappear from the fossil record something like 30,000 years ago. And as we also heard nicely laid out before lunch, there are two competing. Today, there's really just one model of origin of modern humans that are widely hailed, that we come out of Africa, as we discussed. But what is still discussed is when modern humans came out of Africa, Is it so that there was admixture, so that Neanderthals in Europe contributed some genetic variants to Europeans today, and the same went on in Asia? Or is there a complete replacement where people coming out of Africa didn't mix at all with Neanderthals and folks in Asia? So this is a very extreme view, but the good thing is that if you study any particular part of our genome, any particular little piece of DNA, either the right model or the left model must be right. Either there was admixture for that part of our genome, and then if you looked at the Neanderthal, it would be closer at least to some Europeans than to people in Africa or people in Asia. If there was no admixture, then the Neanderthal would be equally distant from people no matter where they live, in Africa, in Europe, or in Asia. So we got a chance to look at this for the first time then in 1997, when we got a chance to extract DNA from this fossil. And this is not just any fossil, it's the Neanderthal from Neanderthal that was found in 1856 and gave its name to this group of hominids. And I should be careful, since there are distinguished paleontologists here, but I think we were very lucky in that the first DNA we could extract was actually from the type specimen that defines what a Neanderthal is, so to say. Because almost every Neanderthal we've since worked on, at least someone has come and told us it's not quite a typical Neanderthal, it looks a little gracile, it looks a little that, but if this is not a Neanderthal, then Neanderthals don't exist, so to say. So we then extracted a sample from the left upper humerus here, extracted under sterile conditions to avoid contamination, focus on a very variable part of this mitochondrial genome, reconstruct cumbersomely with many, many small pieces of DNA, repeated many times, and believe the things that are consistently there in all experiments. And then we can begin comparing this piece of DNA to that in present-day humans. And this is an analysis where we have a few thousand contemporary humans and just look at pairwise differences, human to human, that exist today. And we have a distribution of differences like this with a mean and a mode around 6, 7 differences. This is comparing these humans to this one Neanderthal. The distribution of differences look like this, something 26, 27 differences. For comparisons, humans to chimps here. So clearly the Neanderthals seem to fall outside the variation of modern humans. And it's also the situation that if you look on the range of differences, we don't find some few Europeans that would be more close to Neanderthals than others. The minimum distance to the Neanderthal is the same wherever we look in the world. So we reconstruct the phylogenetic tree for this. We find then that for the mitochondrial DNA, humans go back to common ancestor 100, 200,000 years ago. And something like a bit over 500,000 years ago back is a common ancestor with the Neanderthal. But this was now only a single Neanderthal, so of course the relevant question is, what about other Neanderthals? Could they fall in here? Could there have been admixture for this part of the genome? So there have now been several other Neanderthals retrieved. First was one in the Caucasus, and one from Croatia here. They all, more than 20 Neanderthals, fall together here outside the variation of modern humans. So it's very clear that for the mitochondrial DNA, there is no evidence that Neanderthals contributed anything genetically to us. But we should not forget that if we look in our nuclear genome, our variation is much, much deeper. If you just look in people in this room, for example, many variants we have go back 800,000, 1 million years, so much further than our separation to Neanderthals, something like 400,000 or 500,000 years ago. So even if there was no mixture with Neanderthals whatsoever, it's still true that if we, for some parts of our nuclear genome, trace our ancestry back, some people in this room will be closer to a Neanderthal than other people in this room who go back further here. So, in other words then, we are, even if there was no admixture, are we not that different from the Neanderthals? And this is an attempt to illustrate that. So that means then that what would be very interesting would actually be to study the whole genome, the nuclear genome of Neanderthals. Because Neanderthals are our closest relatives, all categories. We separated from them 300, 400, maybe 500,000 years ago. So we would then be able to say what genetic changes happen in our genome after that split from the Neanderthals, in the last little bit of human evolution. That will be a rather comparatively short list of the changes, and among them maybe some interesting ones that actually is crucial for what makes fully modern human different from other forms. But to do then that, we need to leave the comfort of the mitochondrial genome and go to the nuclear genome, the vast majority of our genetic material. And in the last 2 years, it has been possible to start doing this. And the revolution is driven by technology, as so often in biology, and it comes from new DNA sequencing techniques that make it possible to sequence millions and millions of DNA sequences in just a few hours using new technologies. So what you would then do is make an extract from a fossil. You do a run on your sequencing machine that generates in the order of, say, 50 million DNA sequences, so you get a little database here that you can start comparing to the human genome that we know, to the chimpanzee genome that we know, to bacterial genomes, fungal genomes, and so on. And the first place where this worked was a cave site in Croatia, Vindija Cave, and actually from this piece of bone, from this little piece here, a 38,000-year-old bone of a Neanderthal. The fragments of DNA we then find are really very short. Distribution like this, on average 70 base pair long pieces. So when this first worked 2.5 years ago, we got together with actually the founder of the company that put the first of these technologies on the market, Jonathan Rothberg, and set us a goal of sequencing 3 billion base pairs, so the equivalent of an entire Neanderthal genome, by the end of this year, 2008. And as often a bit regretted actually setting a timeline for this, but it seems that we are almost going to make it, I think. We are here now just about the time to ramp up the sequencing. So if this conference was a few months later, I would be able to tell you much more about the Neanderthal genome than I can now. And actually what we will discuss is a data freeze from May of around 70 million base pairs that we will talk about. But although we then just have a fraction of the total genome of the Neanderthal, we can look at some parts of the genome where we have a lot of data to address some technical issues before we look into the biology. And it's again the mitochondrial genome that exists in many copies, so we have actually by now sequenced the mitochondrial genome of this individual 35 times over. And the issues I briefly want to touch upon is then chemical damage to the DNA and contamination. So damage first. What we can then do is look in all our sequencing reads. Say for this position here, everyone says A. Here, almost everyone says a C, but there are 2 ones that say T here. So we can look at these errors in individual sequences from our consensus of our sequence and see what they are. And the pattern is very clear. It all goes back to modifications of one base C to uracil. And uracil is then read as T. So we have C to T changes. And that's the result of a spontaneous hydrolytic process that happens in the fossil as it lies in the sediments. So our model of how these DNA sequences look is that they are double-stranded, but in their ends here, they have single-stranded pieces where we have lots of this damage C to T. Something like 70% of the C's are T's here, and the ends of the molecule inside it's around 1%. The other issue, which is in a way much more serious, is contamination with modern human DNA. Because as I indicated, we are so close to the Neanderthal, so just from looking at anyone's DNA sequence, it's impossible to tell if that's from a Neanderthal or from a modern human that may have handled the fossil or from us in the laboratory. So we try to limit contamination by working under clean room conditions, and we do assays in the fossil extracts that we start working with. And we can then use these differences that we now know between thousands of humans that have been studied for the mitochondrial DNA and 20 Neanderthals So we can then look in the mitochondrial molecules that we can amplify in an extract, and often it's again very depressing. Often the vast majority is contamination and there are rare Neanderthal molecules in there. Sometimes it's like 50-50, but there are also fossils where actually the vast majority of the human-like DNA comes from the Neanderthal. So we then select those few extracts, and these are quite few. So we to date looked at around 200 extracts from 70 fossils from 6 sites and found 3 bones that are actually almost free of contamination. We then do a few other tricks. We ligate onto the ends of the molecule a little Neanderthal-specific key in our clean room, so that we know when this goes out to become sequenced that this molecule was actually in our clean room and is not mixed up with something after that. And what is now good when we get lots of data is that we can look in our output, in the sequences we actually produce for evidence of contamination. There are now many positions around the mitochondrial genome where the Neanderthal differ from all humans that have been studied to date. So you can look in many, many sequences and start saying, whoops, here is one that looks like a modern human contaminant. So our best estimate of contamination at the moment in our data is 0.5%. We have 9 contaminants among this 1900 sequences. We can also go back to early work 2 years ago, where we actually took our things out of the clean room without this tag. We look on what we published in Nature at the time. There are 10 informative sequences, none look like a contaminant. But as an illustration that you have to be very careful and reevaluate your work when you do science, is that if you actually look at the data from that time, we and others pointed out that there were evidence in the data we produced that it could have been contamination if you looked at the divergence from the human genome and some other features here. Because in another data set we produced, if you compare them, this one looks like it's closer to humans. So indeed, if we go and sequence more in these old extracts, we actually find among 75 sequences 8 contaminants. So we have a contamination rate of something like 10–11% there. So it's absolutely crucial, I think, to do all this work under clean room conditions and have this key here. What we will need, though, is also an assay for contamination from the nuclear genome. And we have to leave the mitochondrial genome. And I think we'll actually be quite sexist in this project. We will focus on male Neanderthal remains, because males will carry only a single X chromosome, as you know. So what we can then look at, we know that any male individual should have only one sequence from their X chromosome. If we find differences there, it has to come from contamination. So we'll actually focus on a male Neanderthal individual for this project. So having then dealt with these technical issues, we can then turn to what we see in the data, actually. So what we have done, as I said, is go around and look for good Neanderthal fossils. And we used the opportunity to actually go outside the classical range of Neanderthals in Europe and Western Asia. The easternmost Neanderthal you will find in a textbook that almost everyone agrees is a Neanderthal is in Uzbekistan. So we also got samples, a child here. We also got samples from southern Siberia here that are so fragmentary, so you cannot say what they really are morphologically. When we looked at their mitochondrial DNA with this assay I described for contamination, we find that they are indeed Neanderthals, and they fall within the variation of other Neanderthals. So this is the one from Uzbekistan, this is the one from Russia. So one insight that has come almost as a side product of this project is that the Neanderthals were actually further east than what we generally think. They were in southern Siberia, on the border to Mongolia and China. So that's why I think an interesting research direction in the next few years is actually go to China and Mongolia and see what were the people there around 30–40,000 years ago. Maybe we'll actually find a Marco Polo Neanderthal one day in China. So what we're also interested in then is finding other Neanderthals useful for this project. The first one was from Croatia here, as we discussed. The other one we use is from Germany, from the Neanderthal site, this specimen still. where a sample had been removed for dating and DNA work earlier, and we were allowed to take another sample a year ago. And the third site is actually very ideal for this type of work. It's a site in northern Spain here, in this mountain here, a cave. It's a fascinating site, a deep cave, where when you go in a side gallery, you find the remains of 9 Neanderthals. Classical Neanderthals around 43,000 years ago that has clearly been eaten by other Neanderthals actually. You have lots of cut marks where muscles attach to the bones, long bones have been crushed to get to the bone marrow and so on. But the good thing is that they excavate there now, so whenever they start finding a bone, they can dress up in sterile clothing that we give them, remove the bone with so little contamination as you can imagine, put them in sterile bags and freeze them inside the cave so that they can frozen to our clean room and then look like this after you've cleaned them. So we have these 3 sites, all around 40,000 years ago. And this is how much DNA sequence we then determined from them in May last year. This is 4 billion base pairs here. Looks very impressive, but you should then not forget that the vast majority of the DNA is bacterial and fungal. I didn't say that, but only around 1% or up to maybe 4 or 5% in the best cases of the DNA in such a bone actually come from the Neanderthal. The rest comes from bacteria and fungi that have lived in the bone after the Neanderthal died. So what we then do is take the Neanderthal sequences, compare it to the human genome, to the chimp genome, find the best places, make sure these are the places that really correspond to each other in the human and Neanderthal genome, in chimp genomes, and start asking questions. And the first question you might want to ask is, well, this divergence from us to common ancestor to the Neanderthal, how far back on this lineage to the common ancestor with the chimp is that? So how far back is this in percent of this lineage? And the answer to that is 12.4%. So if we say 6.5 million years for this point, this means that this is then in the order of 800,000 years ago or so, for the divergence of DNA sequences to the Neanderthal. The population split, of course, is more recent, because if we trace 2 DNA sequences back, they don't go back to the time point where the population split, but into the variation of that population and a bit further back. So to get perspective on this, with also sequence with the same technology, a typical European person, which we take to mean a French person, and that divergence is then 8% back, something like 500,000, a Chinese individual, similar, and some of the deepest divergences that may be in Africa, which is something like 10% here. So the Neanderthal is something like 50% deeper as typical non-Africans, and something like 20–30% deeper than the deepest divergences we know of inside Africa. There are some other parts of the genome that may be interesting to look at. One is the Y chromosome. The mitochondrial DNA we know a lot about, and it's inherited on the maternal side from mother to offspring, and we know Neanderthals didn't contribute that. The Y chromosome is of course inherited from fathers to sons, just the male side of history if you like. And it doesn't recombine most of it, so people, particularly Peter Underhill and collaborators, have estimated a phylogeny for all Y chromosomes around the world. And we find a typical pattern of where the deepest splits go to Africans, and then we find Africans and others here. So what we can now do among our Neanderthal reads is start to look for when we hit these positions that define these deep splits in the Y chromosomal phylogeny. And for each such position, say, well, is the Neanderthal ancestral? Does it fall outside the variation of Y chromosomes in human males today, or inside here? And the answer is that for all such positions, we have found the Neanderthal is ancestral. So unless they carry some very rare variant here, it's clear that the Neanderthal Y chromosome falls outside the variation of modern humans. How deep the split is, we need more data to be able to say. So there are a few other genes that may be of particular interest that we started hitting, and one I want to spend a couple of minutes on is a gene called FOXP2. This gene is interesting because it was found a number of years ago by Tony Monaco's group in Oxford in a family that has a severe language and speech problem. So when one copy of this gene is knocked out in an individual, you have big problems with producing speech. So difficult for you so that naive listeners at least will not understand you, but family members will learn to understand what you say. The evolution of this gene also looks interesting because in a tiny segment of it here, we have 2 amino acid changes in the encoded protein that's unique to humans and differ from chimps and apes and other primates. So of interest was, of course, to start asking, well, are these amino acid changes here that may be of interest specific to humans relative to Neanderthals or shared with them? So a while ago, we now, in our shotgun reads, for the first time hit this. So this is one of the positions that change in amino acids. It's a C in the chimp, an A in modern humans, and the Neanderthals, actually to our surprise, turned out to have an A, just like us. So we went after this more systematically by targeting this piece of DNA in the 2 Spanish Neanderthals from that cave I described, doing lots of controls to make sure the mitochondrial DNA and the Y chromosome and so on is ancestral and really Neanderthal-like here. And it turns out that both these positions in both these other Neanderthals are really derived, just like in modern humans. So it means then that from the point of view of the only gene we know has to do with language, there's no reason to assume that they were different from us. But there are, of course, many, many genes we don't yet know that also have to do with language that we just haven't found yet that could mean that they were different from us. But from what little we know about the genetics of language, the Neanderthals may not have been different from us. So this then means that these changes in FOXP2 were older than the separation from the Neanderthals. There are also actually evidence that something more recent happened in this gene, so there may still be something human-specific here. But what I want to focus on in a few minutes is then, well, these amino acid changes, does it then mean that they are really not very interesting for origins of humans, since they are not different from the Neanderthals? And how can we ask that question? And it's a general question in this field, I think. Whenever we now find something in our genome that we think is of interest for being a fully modern human, how do we ever make our point for that or prove our point, since we cannot do experiments in humans? And per definition, we cannot easily look at animals because we look at human-specific things. Well, my idea about this is that we have to try to use animal models in some form, after all. And in this case, it's actually rather easy, because these 2 human-specific changes are localized to one little part of the gene here. And apart from that, this gene, FOXP2, is very conserved. So if you compare a human to a mouse, there's only one other difference here, a conservative difference in the protein that's encoded. So if we just, in the mouse, exchange this part of the gene for the human here, we essentially create a human gene for FOXP2 in the mouse. And you can do that, of course, by targeting this gene with homologous recombination in mouse stem cells. You can get rid of the selected markers you need here in the vicinity, so you then have a mouse that from its own FOXP2 gene makes essentially the human protein. So then you have the next problem that we found ourselves in front of 2 years ago then. You have this mouse, and you look at it, and how do you test for its linguistic ability then? So you try to speak to the mouse. We did that for 6 months, and nothing much came out of it. So what we then said is, well, obviously it could have to do with almost anything. Since this is a very conserved gene, it must be involved in many functions in all mammals, actually. So we started a collaboration with something called the Mouse Clinic outside Munich in southern Germany, where they do very thorough health checks on mice, much better than what you get when you go to the doctor. So just to give you a feeling for this, they test hearing, bone density, do pathology, muscle strength, startle response, hematology, eye function, blood pressure, EKG, and so on. All in all, they analyzed the mice for 323 different phenotypes. Not all of them independent of each other, many very correlated, but it's still a much better health check than what you get at the doctor. Always comparing these humanized mice to litter mates that have been born by the same mother together with them, but were wild type, didn't have this human form of the gene. And there were only 2 features in which these mice differ from their litter mates that are wild type. The first one I have a not easy time to think about, maybe there are ideas from the audience. And that is that they are slightly more cautious in a new environment. So you measure that by having them in a social group. The mice get to go in and explore an open area. Mice feel more comfortable along the walls here. And you see that the humanized mice here hardly ever in the initial phase go into the center, whereas wild-type mice are bolder. But so this is just a transient thing. For the first few minutes in a new environment, the humanized mice are more cautious. As I said, I have no idea what this is about. The other feature where they differ really knocked me off my feet almost. And that is that they have altered vocalization. To measure vocalization in the pups that are removed from their mother, you have a microphone here and a peep to get the mother to come back to bring them to the nest, and you record that. And we work with people that are then experts in analyzing sonograms of these vocalizations. And there are a number of features you can look at, the peak frequency, the starting frequency, and so on. And there are a number of features here, such as a slope here, where the humanized mice differ from the wild-type mice. And actually the heterozygotes that have one copy of the humanized ones are in between. There's other features here that are the same and others here. So this is very encouraging to me. It sort of supports the idea that this gene, the changes in this gene that happen on the human lineage actually have something to do probably with muscle coordination or so of the oropharynx, the millisecond control we need between our vocal cords, the tongues, the lips to produce articulate speech. And we now have a mouse model where we can begin asking about the neurological background for those changes over the next few years. So what happened with this gene is this change here shared with the Neanderthals. And as I said, there is also something more recent that have happened that we don't know what it is, that didn't change the protein directly. Maybe it's expression or something like that, that we are also actively looking for at the moment. There are, just before we end, the last few minutes, there's 2 more genes I want to mention, or 2 more parts of the genome. One is a huge region here on chromosome 17, which is an example of several parts of the genome where people have suggested that these might have been contributed by Neanderthals into modern humans when they came into Europe out of Africa then 40,000, 30,000 years ago. And this region is special in that there are 2 versions of this gene that exist in modern humans today. There's a deep divergence between these 2 forms that go back almost 3 million years here. It's a very, very deep divergence. What is also very interesting is that whereas one of these versions in humans today have a lot of variation, another one has very little variation, although it's quite high frequency in Europe particularly. So since it has little variation, it suggests it's a recent change that spread quickly. So it looks like it had been under positive selection. And in fact, there is a study in Iceland that shows that women that carry this version of this part of the chromosome has higher fertility than others. So there seems indeed to be positive selection going on here. And since this occurs, particularly in Europe and Western Asia, it has of course been suggested that this comes from Neanderthals into modern humans and is positively selected. So we now got a chance then to look at this in this first Neanderthal sequence. So this is a number of positions in this huge segment, 900 base pairs. And we see the frequency difference, 100% frequency difference in the yellow ones here between one version, and this is a positively selected one specific for Europe. And if you look at the basis in each of these positions, you will see that each position here is actually carries the common variant that we find all over the world, not the one we find in Europe. So the 2 chromosomes in this first Neanderthal from Croatia we sequenced clearly does not carry this variant that is suggested to come from Neanderthals. Of course, we will have to look at more Neanderthals as time goes, but I would like to point out that we also find this in Africa, although at very low frequency. And I'll come back to that in a second. So another gene where the story is very similar is microcephaly on chromosome 8. This gene is quite interesting because it's a bit like FOXP2. When it's mutated in humans, you have a very small brain, a chimpanzee-like brain, but of normal structure and just a mild mental retardation. If you look on the variation of this gene in humans today, it's also quite old, go back 1.7 million years or so. And again, there is one version with little variation, but that is quite frequent and exists particularly outside Africa here. So it has again been suggested by Bruce Lawn that this is contributed by archaic forms of humans such as Neanderthals. So we have now been lucky enough to hit the diagnostic position in this gene where the derived version, the one that exists outside Africa, has a C, others have a G. So this is the positions where humans, and most humans have a G, and the Neanderthal turns out to also have a G, to not have this version that exists particularly in Europe and Asia. Again, this variant exists also in Africa. It's not absent in Africa. So I think at the end of the day, for most of these things that have been suggested to come from Neanderthals into non-Africans that exist today when they came out of Africa, I think instead what has been going on in most cases is actually that when modern humans originated somewhere in Africa, probably as we heard in southern Africa, they spread first across Africa and incorporated some deep old variants here that they took with them when they left Africa, and just from drift in small populations, some of these came to high frequency in non-Africans today. We can also now start asking much more directly what happened when modern humans came out and met Neanderthals in Europe and Western Asia. Did they mix or not? In a more statistical way all over the genome. And to do that, we make use of what I started out by talking about, that if there was no admixture across the genome, then a Neanderthal would be equally distant from people in Africa today as from people in Europe. If there was admixture, then Neanderthals would on average be a bit closer to Europeans than they would be to Africans. So to ask that question, we need to compare the Neanderthal to variation we find today in humans. And we need to look at that variation in a very objective way, because there is a problem that most work, not only as we heard before lunch in paleontology, have been done in Europe, but most genetic work has also been done in this country and in Europe in people of European descent. So if we just look at what we know about genetic variation, it looks like Europeans are most variable, but that's just because we've studied them more. Overall, as I said, we know Africans are more variable. So in order to look objectively at differences, we decided to just compare one single African to one single European of European descent, and just look at positions where these two individuals happen to differ from each other. And then we have these two individuals here with a difference, and we take the Neanderthal, and we say on average across many such positions, how often is a Neanderthal as the European, how often is it as the African? If it on average is closer to Europeans, that would argue for gene flow. If it's equally distant from them, it would argue like the Neanderthals, an earlier divergence all over the genome. So just to show that this really works, if we take another European today and sequence with the same technology as we used for the Neanderthal, we find that in 63% indeed one European looked like the other European rather than the African in 63% of such cases. When we now take our Neanderthal data and do the same analysis, it looks like this, almost dead on, 50.2%. So this 0.2% there is compatible with an admixture or contamination in our data of 1.5%, but it's also compatible with 0, as you see here. So as we now go on and do the whole genome, we'll have much more power to detect even a small contribution. But it's very clear from what we see today that if there was a contribution from the Neanderthal, it is very, very small indeed. So to wrap up then, the big picture all over our genome is clearly one of replacement. Could have been some little contribution, but if it was quite small. So to end then, I think that is quite good news for this Neanderthal genome project actually, because if we think for a minute about why are we doing this, why are we putting so much effort into getting a Neanderthal genome, there are really 3 reasons for it that I want to mention. One is quite obvious, and we already discussed it, that is that there is this quite long lineage from the separation to the chimp here, 5 to 7 million years or so. So having the genome of the Neanderthal would be able to divide up all the genetic changes on the lineage in those that are very recent and unique to us, whereas those that are older. And we already know that this is around 12.4% of the differences here. So we will have the human genome we already have, the chimp genome, we have 35 million positions where they differ. We'll have one Neanderthal and find the 12% of positions where the Neanderthal looks chimp-like. The second big goal we have is a little more subtle, and it makes use of this thing we mentioned that the variation among humans that live today is so big so it goes back to before the separation from Neanderthals, goes back before 300,000–400,000 years here. So that of course then means that in an average part of the genome, if we look at positions where we vary today, we will know which is the old variant because we have that in the chimp. So in this case, the T is a new mutated variant, so to say. In quite a few such positions where we vary, the Neanderthal will actually have the new, derived, mutated variant that's not in the chimp, in average parts of the genome. But in positions where something interesting happened recently, where there was a positive selective sweep in humans since we separated from the Neanderthal, in such positions, of course, it means that in that part of the chromosome, something happened, some variant that spread to all humans today because it had an advantage. There, of course, the variation today is per definition more recent than the separation to the Neanderthal. So in such a region, the Neanderthal will always look chimp-like. So what we will do is use the 2 or 4 million positions we will know about where humans vary today. We know the ancestral old condition from the chimp. We'll have a single Neanderthal, and we'll look in a window. And in any average window, in some positions, humans, the Neanderthal will have a new mutated variant, in other, the chimp one. We'll slide that across the genome and look for regions like this, where suddenly Neanderthal looks all chimp, because there something interesting seems to have happened, something positively selected since we separated from the Neanderthal. So our goals is this catalog of changes that are recent, shared among all humans today, but different from the Neanderthal, evidence of positive selection, and eventually, of course, also looking for Neanderthal-specific features, although that will really have to await a much better Neanderthal genome over the next 2 or 3 next years. So with that, I should end and just say that what I presented, the production of the sequences, is an intimate collaboration with 454 Life Sciences in Brantford, Connecticut, Mike Ronan particularly, that worked with us in Leipzig on this. I should say that this is a close collaboration also with the paleontologists that excavate the fossils, particularly with the team in Spain that excavate El Cedron, and also people in Croatia. Some of the analysis I showed of the SNP analysis have been done by David Reich and Jim Mulliken at the Broad Institute in Boston and the NHGRI. And I should also end up by saying that we will, as you notice, produce a lot of data over the next few years. So we are looking for people to help us with analyzing all of this. So if you're interested in bioinformatics or in genome sciences, please talk to us. Thank you very much.
Could I ask the other members of the panel, please, to come up and join us at the table here? And we'll begin the Q&A session in perhaps 5 minutes here.
Gentlemen,
yeah, we are on.
ladies and gentlemen, could you take your seats, please? We'll begin the Q&A session here in just a moment.
They're handing out question cards. Okay.
Whoops.
Okay.
Oh. Okay. Do you want to hear me?
The problem is that when you turn around...
Yeah.
Okay. All right, ladies and gentlemen, let's take our seats here and we'll begin. We'll ask other members of the panels if they have questions or comments about Dr. Pääbo's talk here. Dr. Dunbar.
I'll just go back to the FOXP2 stuff, because that's clearly really very interesting. But when it first came out, it was kind of trumpeted as the gene for grammar. But what you're clearly showing with the mouse stuff is that it's actually something to do with the capacity for vocalization, which kind of then makes it very interesting in the context of whether Neanderthals had language as we know and love it, or whether they had some other form of vocal communication. And this has raised one of the possibilities that the precursor to language, before full-blown modern human languages came in with grammatical structure, was a kind of vocal chorusing, the sort of singing Neanderthal model. Do you have any comment on that possibility?
Not really any comment. I mean, I think that, yes, when this gene was first described, it was talked about as a grammar gene or the language gene, and as I understand the consensus now among the people who study it, that's really based on rather bad clinical work. That the problem also in the family is one of articulation, is a sort of basic problem of muscle coordination in the oropharynx. And it seems then that these human-specific substitutions also have something to do with that. It may be that FOXP2 does other things also that has to do with communication. It's actually fascinating that if you look in birds, in songbirds, if you knock down this gene, interrupt it, you disturb vocal learning in birds when they learn their song. And I just learned the other day that the gene has now been found even in octopus. And it's actually expressed in the neurons where octopus make color signals to mates or aggressive signals. So it seems to be incredibly conserved and have to do with communication in animals.
Tim?
Dr. Van Hoystein.
Just to add to that, I wondered about this, Stephen Mythen's book that maybe you'd refer to now. Does it even make sense then to speculate about proto-language, for those who are interested in the evolution of language, what might have been the case in terms of Neanderthal, in terms of either music or proto-language, or are you cynical about that, other than saying that it might, in the sense that you just said, indicate some form of communication?
In a way, I try to really stay away from speculation. And I say that from the little we know, there's, from the point of view of these amino acid substitutions, there's no reason to say they were different from us. As I said, there is genetic evidence that something else had hit this gene. There's some evidence of a recent selective sweep that's actually different from the amino acid changes. We didn't know that when we first published it, but it's now obvious from the data. So there may still be something there, but we have to find it first and define it. And before that, I wouldn't. It seems obvious to me that the Neanderthals at the time must have had communication that was very similar to modern humans. They lived in social groups, they took care of injured people, etc., etc.
How about, if I may ask one more quick question? I was interested in your conclusion about the Alcidron cave. It seems like a really strong conclusion to go from scratches on the bones to cannibalism. Did I understand you correctly? Neanderthals eating Neanderthals?
Yes, absolutely, yes. It seems, I understand from the paleontologists that it's more and more common now when you look for the traces of defleshing. And there's, of course, also discussion, is this some ritual that has to do with burial or so? Or is it actually consuming your fellow Neanderthals? Being in that cave, although I'm not an expert, I do think it looks rather more like eating your neighbors. It's a group that seems to have been washed into the cave at one time. There's 2 infants, 2 adolescents, and adults. The hands are in one part of the cave, the rest is somewhere else. It looks pretty grim to me.
I was going to point out that it's not unique to Neanderthals. There's a site on the coast of South Africa called Klossi's River Mouth that has a series of hominid remains. These are Homo sapiens that date around anywhere between 70,000 and 90,000. And there's a very high frequency of stone tool cut marks on the human remains as well. And in fact, the frequency of cutting is higher than it is with the antelope bones that are in that collection. But it's always difficult to go from that pattern to an interpretation of what it means. Is it cannibalism for the purpose of food, or is it some more ritualized bodily defleshing activity? And at least with the South African material, we don't have a big enough sample to say one way or the other. But the pattern of stone tool modification of human remains, cut marks, and hammerstone percussion marks is fairly common in the Paleolithic.
Yeah, but another question might be, who's eating them? Is it other Neanderthals, or might it be another group of hominids?
It's
Homo sapiens.
clearly Neanderthals at that time. At least as far as we know, there were not yet modern humans at that time in Iberian Peninsula.
Dr. Feldman.
Do you think it's fair to use mutations like FOXP2 and microcephaly, which have huge developmental impacts on the human carriers, to ask questions about evolution at the structural level that we're all interested in?
Well, so I think that these genes are found because mutations in them just give disease phenotypes in humans.
Very, very serious diseases. So.
Depends on how serious. I mean, articulation problem, how serious on those two. But this is one way of finding genes that are involved in these pathways, right? Of course, we can now go and look for the targets of FOXP2. What I didn't say is FOXP2 is a gene whose function is to turn on and turn off other genes. Mm-hmm. So one can, of course, look for the targets for that gene and look also for special evidence in those. But it's perhaps a limitation also of where we are in human genetics today. If we are interested in genes in language, for example, we're really to a big extent limited to studying pathologies of language to find these things.
But they often turn out to be pathologies of other things too. And that's some of the problems, that they're not just pathologies of language. They're developmental abnormalities as well.
There's a related question that goes along with this. This is an internet question here that came. How do you think that these genetic modifications that resulted in, say, the microcephaly gene have been able to persist so long and are so far-reaching, considering that the phenotype results in a smaller brain?
Well, so that gene, the mutation that causes the smaller brain size, is very rare and occurs just in rare families in Pakistan and in Sinai. The gene itself is necessary for the development of the brain, and it's conserved in all of us. So it's just this mutation, and that's very, very rare.
Okay. Do any other panelists have any other observations?
I just had a question. What do you think the potential is for finding ancient DNA that dates to the time period of the early moderns in Africa?
So I think from the point of view how old it is, it should be possible. What makes me skeptical is the warm climate. It's very clear that the warmer it is, the worse it is for DNA preservation. So we have tried some Neanderthals in the Middle East. None of them have worked. We tried some animals from Blombos Cave, actually, and didn't get anything. So maybe it's one finds very deep cave site with cold temperatures. But,
are there any other conditions, for example, maybe in bogs or anything like that, that helps? Or is it all temperature mostly?
So another important factor is pH. Acidic conditions are very bad. So most bogs, say in Europe, are acidic.
Okay.
In the Yucatan Peninsula, for example, or in Florida particularly, there are alkaline bogs that are quite good for DNA preservation. But these are the rare cases.
you give me hope then that perhaps in Siberia we might be able to extract DNA that will help unravel the issues of the people of the Americas?
Yes, I think permafrosts are excellent conditions. And there are mammoths, for example, where up to 70% of all the DNA in the specimen is actually from the mammoth.
Mm-hmm. Good.
Now here we have a question from the audience. Is when you look at the chimpanzees and the modern human and talk about positively selected genes, why did the ancestors of the chimps not also have, why weren't they selected for that positive trait as well? Is it the assumption that the chimp has not changed in the last 700,000 years?
Mm-hmm. No, so actually there was lots of positive selection in the chimps too, only that was chimp positive selection. So if you look for just evidence of positive selection on the chimpanzee lineage and the human lineage, there's even some evidence, although it's probably not so safe conclusion, that there have been more positive selection in the chimpanzees than in humans.
There's another question. What specific characteristics actually determined or distinguished Homo sapiens from their predecessors? Was it all the genomes and DNA or the other distinguishing characteristics?
So that's probably more a question almost for a paleontologist. Of course, what our million-dollar question in our field would be to find the few genetic changes that actually made a difference. Then we can, of course, discuss culturally and behaviorally what are these major things that distinguish us from archaic forms of humans, from Neanderthals or the common ancestor of humans and Neanderthals. It's one thing that tends to strike me, for example, when we look at the presentation this morning, is that these earlier forms of humans, as you said, left Africa 2 million years ago, the first. And they never came to the Americas, never to Australia. There's actually no evidence they ever crossed water where you don't see land on the other side. So they were quite sane. Then comes early modern humans out of Africa 40,000 years ago. And in those 40,000 years ago, we go to the Americas, Australia, and to every little speck of island, right? We go to Easter Island. And now we go to Mars. So there's some craziness there.
Robert, may I? Just the glass.
There's a question about your FOXP2 mouse that you have. We saw the mouse that had the human FOX2 gene variant. Is there a FOXP2 mouse knockout gene that doesn't have that gene, and what does this look like?
Yes, so that's a very good question. If you knock out the gene, and we and others have done that, in the homozygous form, where you have both copies destroyed, you actually don't survive at all. You die shortly after birth. If you have one copy knocked out, like this family in Britain, or in the mouse, you have a number of, in the mouse, you have a number of muscle coordination problems, and problems with lung and many physiological issues that happen. You're actually quite sicker as a mouse than as a human.
Okay. Here's a question too. It says, the Basques in the mountains near Spain are claimed to have a unique genetic history. Do you have any information on how they may be related to Neanderthals?
Yes. You should be careful with you say Basques had bombs and things like that. They are actually just as modern humans as all of us are. There's no special relationship there whatsoever.
Svante, there is some suggestion that they are the remnants of the kind of original modern human occupants of Europe who are replaced by the Indo-Europeans, or nearly overrun. I mean, is that still
I'm
the case?
not an expert on that, maybe, Mark.
Yeah, there are two lines of evidence. One is that their language doesn't fit on any language tree of known languages.
Is the nearest language is Mongolia or something?
In somewhere close to Siberia, Lake Yenisei. The other thing is that They're the only population on the planet that has a higher Rh negative frequency than Rh positive. So there is something strange about their evolutionary history. But it may be due to, simply due to their isolation in the Pyrenees, but nobody knows beyond that.
Okay. Well, here's one final question of practical significance for us. How useful and valid are the DNA swabs presented by various websites?
Oh, I want to know that.
Yes, so I think that, of course, it can be interesting to relate, for example, your mitochondrial DNA to that tree of all the mitochondrial DNAs there. I think you should not forget, if you do that, for example, that that's a tiny, tiny little fraction of your genetic inheritance. There are chat groups at these sites where everyone in the same related mitochondrial DNA gets in and talks to each other, and it seems to be ridiculous to me. It seems to be just like astrology is related to astronomy, this form of genetics is related to what we do.
Can I answer that question?
Yes, Dr. Feldman.
There are three companies that are competing at the moment for that market that I regarded as legitimate. And they each has two components to it. One I call Recreational Genetics, which is where you ask what places on the planet your ancestors might have come from. The other one is not so recreational, and that is the company has huge databases, and they look at your DNA, not just the mitochondrion, not just the Y chromosome, but lots of the rest of your genome, and has a list of the variants that may predispose you to certain traits. And there are about 12 disease traits which are currently accepted by the genetics community as having a single change in the DNA, which is a good risk marker. The risk may only be an added 2 or 3%, but that's something that you might want to know about. So those are the two things that the companies do, and the good companies on their website say exactly what they do. And they're very clear about what you can expect from the information and how to use it.
Well, at this point, I think we should break for a little coffee. Remember, this can be found in the Forum, which is the building just to the north of us here. We'll assemble here once again at 3 o'clock.